Assalamu alaikum and welcome everyone to our molecular biology ASCP preparation series. The MCQs we'll discuss today and in the upcoming season have been carefully collected from several years of past exams to give you the best preparation possible. I'm Dr. Lai Abdalati and I'm really excited to share my knowledge and experiences with all of you through this series. So let's dive in together. In our previous lectures, we completed the multiple choice question related to the nucleic acid chemistry and basic molecular theory. All right. Today, we'll move to cover the first part of biochemical reagents, DNA and RNA polymerase enzymes. Now let's focus on the question related to the DNA and RNA polymerase enzymes and review molecular biology ASCP questions from the previous exams. So the first question we have today is, which of the following is the main function of DNA polymerase? DNA polymerase is enzyme responsible for synthesizing new DNA strands by adding nucleotides to pre-exciting strand using a DNA template. So the correct answer here is DNA synthesis. Okay, let's move on. Okay, the second question is, what is the role of DNA polymerase 1 in prokaryotic cells? Okay, let's see the correct answer. DNA polymerase 1 removes RNA primers from the Okazaki fragments and fills the resulting gap with the DNA nucleotides during DNA replication. So the correct answer here is removes RNA primers and fill gaps. All right, let's move on. Okay, next question. Which of the following activities is associated with DNA polymerase 3 in E. coli or prokaryotic? Actually, DNA polymerase 3 in E. coli has 5' to 3' polymerase activity, which is essential for synthesizing new DNA strands. Okay, so the correct answer here is 5' to 3' polymerase activity. Okay, let's see the next question. What is the primary function of the proofreading activity of DNA polymerase? The proofreading activity of DNA polymerase corrects errors by removing mismatched nucleotides and ensuring accurate DNA synthesis. All right, let's move on. Okay, next question. Which of the following is the characteristic of eukaryotic DNA polymerase delta? Actually, DNA polymerase delta in eukaryotes has the high processivity and proofreading activity, okay, making it essential for lagging strand synthesis. So the correct answer here is high processivity and proofreading activity. Okay, let's move on. Okay, next question is, which DNA polymerase is primarily responsible for mitochondrial DNA replication? DNA polymerase gamma. DNA polymerase gamma is responsible for replicating mitochondrial DNA, ensuring the genetic material in mitochondria is accurately copied. All right, let's move on. Which of the following DNA polymerase lacks 3' to 5' exonuclease activity? Which one of them? The correct answer is DNA polymerase alpha. DNA polymerase Alpha, as we know, lacks 3' to 5' exonuclease activity, all right? And is primarily involved in initiating DNA replication by synthesizing RNA primers. All right, let's move on. Okay, the next question here. Which enzyme works in conjunction with the DNA polymerase to ensure high fidelity DNA replication? All right, the correct answer is DNA primase. DNA primase synthesizes RNA primers, which are necessary for DNA polymerase to start what? To start DNA synthesis, ensuring high fidelity during DNA replication. All right, let's move on. Okay, next question. What is the function of 5' to 3' exonuclease activity of DNA polymerase 1? The 5' to 3' exonuclease activity of DNA polymerase 1 removes RNA primers from the Okazaki fragments during DNA replications. So the correct answer here is removing RNA primers. All right, let's move on. Okay, next question. Which of the following statements about DNA polymerase 3 is correct? All right, the DNA polymerase 3 is responsible for synthesizing both the leading and lagging strands during DNA replication in prokaryotes. All right, let's move on. Okay, what role does DNA polymerase beta play in eukaryotic cells? Actually, DNA polymerase beta is involved in the base excision repair, okay? A mechanism to fix damaged DNA bases in eukaryotic cells. So the correct answer here is participating in base excision repair. The next question is, which of the following is the feature of DNA polymerase epsilon? All right, actually DNA polymerase epsilon has the high processivity and proofreading activities, which are essential for synthesizing the leading strand in eukaryotic cells. All right, let's move on. Okay, the next question. Which DNA polymerase is primarily involved in the synthesis of RNA primers? Correct answer is DNA polymerase alpha. DNA polymerase alpha is responsible for synthesizing RNA primers during DNA replication initiation in eukaryotic cells. All right, let's move on. All right, the next question. Which of the following describe the function of DNA polymerase 3 sliding clamp? Okay, the sliding clamp of DNA polymerase 3 enhances processivity by ensuring the enzyme remains attached to the DNA template during the synthesis. All right, let's move on. All right, the next question. What is the role of DNA polymerase 1, 3' to 5' exonuclease activity? The 3' to 5' exonuclease activity of DNA polymerase 1 proofreads and corrects error during the DNA synthesis by removing mismatched nucleotides. All right, so the correct answer here, proofreading, and error correction. Let's move on. All right, the next question is, which of the following DNA polymerase is the most sensitive to acetylcholine? All right, the correct answer is DNA polymerase alpha. DNA polymerase alpha is highly sensitive to acetylcholine because it plays a key role in DNA priming and lacks proofreading abilities. All right, making it easy target for inhibition. So the acetylcholine interferes with the DNA synthesis by blocking nucleotide incorporation, thereby disrupting replication initiation and elongation. Okay, let's move on. All right, next question. Which DNA polymerase is essential for lagging strand synthesis in eukaryotic cells? The correct answer is DNA polymerase delta. DNA polymerase delta is essential for synthesizing the lagging strand in eukaryotic cells by extending Okazaki fragments. Okay, let's move on. All right, the next question. Which of the following is true about DNA polymerase gamma? DNA polymerase gamma replicates mitochondrial DNA, ensuring the genetic material in mitochondria is accurately copied. So the correct answer here is replicate mitochondrial DNA. Please don't forget to subscribe to our channel to receive all the latest lecture. Press. All right, let's continue. Okay, which DNA polymerase had a unique thumb, palm, and finger domain architecture? All right, the correct answer is DNA polymerase 3. Let's move on. All right, let's see the next question. What is the role of DNA polymerase 4 in prokaryotic cells? All right, DNA polymerase 4 is involved in translesion DNA synthesis, allowing DNA replication to continue past damaged site in prokaryotic cells. All right, let's move on. All right, let's see the next question. Which DNA polymerase is primarily involved in the synthesis of the leading strand in eukaryotic cells? The correct answer is DNA polymerase epsilon is the primarily responsible for synthesizing the leading strand during DNA replication in eukaryotic cells. All right, let's move on. All right, the next question is, which DNA polymerase is involved in both DNA synthesis and repair? The correct answer is DNA polymerase beta. DNA polymerase beta is involved in DNA synthesis and repair, particularly base excision repair in eukaryotic cells. All right, let's move on. All right, the next question. Which of the following is a characteristic of DNA polymerase eta? All right, DNA polymerase eta is involved in translesion DNA synthesis, allowing DNA synthesis to continue past this lesion. So the correct answer here is involved in translesion DNA synthesis. All right, the next question here. Which enzyme is responsible for synthesizing RNA from a DNA template? All right, the correct answer here is RNA polymerase. RNA polymerase catalyzes the synthesis of RNA from DNA template during DNA transcription. Unlike DNA polymerase, it doesn't require a primer to start synthesis. All right, let's move on. Okay, let's see the next question here. In prokaryotic cells, which RNA polymerase transcribes all type of RNA, including messenger RNA, ribosomal RNA, and transfer RNA? Actually, prokaryotes use a single RNA polymerase enzyme to transcribe all type of RNA, unlike the eukaryote, which have only 3 different RNA polymerase for different RNA types. So the correct answer here is a single RNA polymerase. All right, let's move on. All right, the next question here. Which subunit of prokaryotic RNA polymerase recognizes the promoter sequence? The correct answer is the sigma subunit. Actually, sigma subunit of prokaryotic RNA polymerase recognize and bind to the promoter sequence, initiating transcription. After transcription begins, then the sigma subunit is released. All right, let's move on. In eukaryotes, which RNA polymerase transcribes messenger RNA? All right, the correct answer is RNA polymerase 2. RNA polymerase 2 is responsible for transcribing messenger RNA in eukaryotic cells. All right, let's move on. Okay, next question. Which of the following is the function of RNA polymerase 1 in eukaryotic cells? Okay, the correct answer is transcription of ribosomal RNA 28S, 18S, and 5.8S ribosomal RNA. All right, which are essential for ribosome function. Let's move on. All right. Which RNA polymerase is responsible for synthesizing transfer RNA in eukaryotes? All right. The correct answer is RNA polymerase 3. All right. Let's move on. Okay, that's all for today. Please don't forget to like, comment if you have any questions, share and subscribe to our channel to receive all the latest lecture. Okay. I also encourage everyone taking the test to share your question with us so that everyone can benefit. All right. That's all. Salam.